When we dock, for example, if we have a protein and the ligand, what are the various aspects we need to think? There are two different aspects, right. These are the major limitations. One is the search algorithm. Right. What is search algorithm?
It will find the search
poses, right, because the protein can form large number of poses. Right. You know the active sites, even the change of the diagonal angles, right, you can see large amount of conformations. So, how to see which one is the best one, so that the ligand can interact. And the second aspect, the scoring function. When your ligand interacts with a protein, how to define or how to quantify this ligand 1 interacts better than ligand 2. So, we need to have a scoring function to decide the binding affinity, right, between the protein as well as the ligand. So, two aspects, one is the pose and the second one is a score, right, which give you the strength of the binding, right. Then how to do this? If you look into the search algorithm, there are various number of search space. If you take the protein ligand docking, right, it involves many degrees of freedom, right. What are the various degrees of freedom when a protein interacts with the ligand? You can see the translation. How many degrees of freedom for the translation? 3 degrees of freedom, right, because you can see x, y and z, 3 degrees of freedom. So, likewise you can see 3 rotations, so totally we have 6 degrees of freedom, right. Then if you see the conformational degrees of freedom, right, because you have the rotations, right, you can see the conformational changes, there are several conformational changes depending upon where, how many places where we can have the rotations. Then if you look at the solvent also may play a significant role in the protein ligand geometry. So, in this case we need to have a large search space. So, then if you need to derive a search algorithm which generates various poses and the different orientations, right, for any particular conformation of a molecule at the binding site. So, it has to generate various orientations and it has to cover the whole search space. For example, if you see simple amino acids, right, and if you have one rotation they can put 0 to 360 degrees. If the two, one will rotate, second will also rotate, right, you can systematically do it, right. So, it takes long time, right. So, there is a trade off between time and search space coverage, right. If you have more time then you can search more space, if you less time you will get less search space. So, in this case we need to have a smart algorithm, right, to get different search space and various poses, right. So, that also depends upon the type of docking what you want to do. Sometimes if you know the ligand, as I discussed earlier, if you know the protein and if you know the ligand, right, in this case the docking is simple, right. Here thus we use the rigid docking. In this case protein is treated as rigid, right. We have the pose, protein pose, right. So, we do not make any changes in the protein pose, we keep the pose as it is. Then the ligand we make different orientations, different conformations we can make, right, and for each conformation, right, we can treat ligand as they also rigid and the protein also rigid and you can see the docking. So, we get one energy. So, ligand conformation also we can decide. So, make the different conformation based on the translation rotational degrees of freedom, right, and you can see how they fit, right, with the protein, that is called rigid docking. In this case protein is fixed, if it is one ligand that is also fixed, but ligand we can make different conformations and each conformation, right, conformation you can get this scoring. Okay. Then the case of flexible docking, what is flexible docking? In this case name itself tells it is flexible, right. In this case you can change the conformation of the protein at the active site, you can make various conformations, right. In this case it takes enormous time, we have the different conformation of this ligand and also we have different conformation of protein. Compared with ligand and protein, which takes more time? Protein takes more time, because ligand takes less time because it is a very small molecule and there are many rings. So, in this case there is not much rotations, but if you take the proteins, right, even each amino acid residue, right, even the 5 residues in the active site, right, there is several degrees of freedom. So, in this case there can make many conformations, right. In this case we need to generate an algorithm, we need to have an algorithm which can sample all these poses or all these conformations. So, there are various types of algorithms, we can systematically search. For example, if you have 3 rotations, right, and we systematically go with 1 degrees, how many times you have to do with to cover all the space? For one case it should go 360 degrees, second also 360 degrees, or 360 into 360 multiplied by 360, right, 360 into 360 360 times you need to do to complete the entire space. Or you can do, instead of 1 degree you can use 5 degrees or 10 degrees, systematically you can do, okay, instead of 1 you can use 10, then 360 you will get 36 times, 36 into 36 into 36 with the 3 rotations. If you enormous, if I 4 or 5, then automatically the number will increase, but it is systematic, but it is very exhaustive, right. We can determine the exactly which conformation fits well with the ligand. If we have one ligand, change the conformation and fit it and get the scoring. You can see these are the possible cases, and we can exactly determine, okay, this is the pose where the ligand can interact with the particular conformation of the protein. That depend upon the granularity of the sampling, right, how much times you want to do, right, what is the deviation, the how much degrees you want to change, right. So, that depends on the number of times you need to do the searching, right, and also it is feasible only for low dimension problems. If there are 2 cases we can do it, 3 rotations we can do. If we have many rotations, then it is not possible, because it is very time consuming, you can do that, because you can see the various degrees of freedom. So, in order to avoid that, right, for example, if you want to systematically do that, and randomly also you can do, right, 1 then 10 20 50 60. If you look at this stochastic search, randomly you can add, change and find, okay, this is possible or not, second you do it and then see is possible or not. Within these 2 you can decide the third one, this third one could probably lie on this place, then based on the 3 you can decide the fourth one. So, in this case, instead of doing millions of times, you can reduce it thousands of times, right, this is called the stochastic search. If you look into these results between systematic and stochastic search, right, you cannot find much deviation, but of course there is a deviation, but not much difference between the poses obtained with systematic search and the stochastic search. So, here this is random and the outcome varies, because if you use 100 conformation, we will get some data, get the binding. If you use another 100 close to this, not be the same, they will be different. So, depending upon the sampling you do, depending on the space you walk, that will give you the outcome, and the outcome varies, right, and you can repeat to improve the chance of success, right. For example, instead of 1 million times you do 10,000 times, if the results are similar to the 1 million times, then that is fine. If not, you can change another chance, right. Also among these times they can choose, okay, 1 and 3 are not good, so we will go to the fourth one. Fourth one is in between 3 and 4, then fifth one you can decide, okay, we can assign some other conformation between these 4 and 5, right, they can do that. And here you can, it is also feasible for higher dimension problems. If you have several rotations, there are also possible, right, to map different conformations in the case of this stochastic search, fine. So, the difference between systematic search and stochastic search are, what are the differences?
So, it is random.
This random in the case of stochastic, the exhaustive in the case of systematic. Systematic we can get the exact pose, okay, this is the one the ligand prefers to bind, but in the case of stochastic outcome will change, and the systematic will depend on the granularity of sampling, right, what is the angle you require to change, right. So, here this will repeat to improve the chance of success, you can change again, and the systematic is feasible only at low dimension, right, but here the stochastic you can go with the higher dimensions, right, mainly for example Autodoc is about 40 dimension search. So, there are various algorithms have been proposed, right, to do the sampling, right, for the case of stochastic search, right, simulated annealing, genetic algorithm, taboo search, right, hybrid global local search and Lamarck GA algorithm, so on. So, various algorithms have been proposed, right, to do the stochastic search. So, I will explain only 2 of them. First one is simulated annealing. Here this is mainly based on temperature. So, the assumption is when we have this high temperature, so the molecule have more degrees of freedom. In this case we have lot of conformations, but all the conformations they will not pass through this. If you take any scoring function, it will not pass through the scoring function. In this case most of them are rejected. Then go with the accepted ones, then we decrease the temperature. When you cool down, you can see there is less degrees of freedom. In this case you can reduce the conformation sampling space, and finally at lower search you can lower temperature you can get less number of sampling. So, do the local search and you can identify, right, the probable conformations, right, using this simulate annealing method. And the another one, this is widely used genetic algorithm, right. The genetic algorithm is a stochastic algorithm to sample the data, right. They mainly used in the global optimization issues. So, what they do, first take a pose, right, initial population is generated, the randomly generated initial population. So, each chromosome represents a pose. So, in this case you can have a random number of many poses, right, that generate the binding sites. Then you check there are some scoring functions based on torsional angles and the other energy potentials, right. See what are the locations of high scoring, take the high scoring ones and you merge these high scoring poses, right, and to generate a new child. So, take randomly generated poses, some of them are having high score, some of them are having low score, and merge this high scoring to generate a child pose. Then the child pose they do some mutations, they can change the torsion angle or then check the orientation. So, make new conformation and then see whether the score increases or decreases. Then if it is increases, then you make the these new child ones with the previous ones original pose to make next generation. Do it again and again and again, right, up to the n generations or n significant improvements is observed. In this case you can identify a high scoring pose. So, starting with the several random conformation, so random poses, you get the one with the high score, then merge the high scores and form the child, then you do the mutations. Mutations means the change in torsion angles and the rotations and find this scoring, then again the fuse with this the high scoring ones, and finally we continue unless we get these significant improvements, right. Then you can identify these pose. So, likewise there are various other algorithms available in the literature to search, right, the conformation space and see what are the probable conformation space for the ligands to interact at the protein site. Fine. So, this is the one aspect. What are the two major aspects in docking? One is sampling
and scoring,
and the scoring function, right. What is the meaning of scoring function? What information you can get from scoring function?
So, in different binding pose, which one is the best?
Best one, right. You can see it is accurately calculate the binding affinity. So, for example, if you have a set of compounds or you do the virtual screening, there is select some compounds from a pool of compounds, it can identify the actives. So, then also it can rank the actives. If there are 10 compounds, you can use this scoring function, it will give the values for all the 10 and see the best ones. You can rank the actives in terms of this affinity, right. Then also you can score the poses of actives, right, higher than poses of inactive, right. For example, you have the 100 actives and 1000 inactives, and if you have scoring function, you can make a scoring function in such a way that the poses of the active should be high score compared with these inactives in the case of virtual screening. Likewise, you can adjust the scoring functions, then this scoring function can be useful to identify the actives compared with the inactives, right. So, then how to generate, how to develop the scoring functions, right, because there are various ways, right. The one is simple one, we can say that if you have a protein and you have a ligand, we need a scoring function. Generally if you talk, what do we expect if we have a protein and the ligand to interact? They shape complement, they should interact, right, they should have in the proper position. So, where we have the convex or concave in the protein, we should be opposite in the case of this ligand, then they have good interactions and then they can make the complex with the tight binding. So, one is a shape and chemical complementarity score, so offer their complementarity. Then you can do some empirical scoring depending upon the known structures, they can calculate the interaction energies. and from that you can check with the known values, activity values, right, and then see some empirical scoring, right. Then we can see the force field scoring, that depending upon the force field interaction energies, you can calculate the interaction, and using the interaction whether you can see okay this could be the probable ligand, right, probable potential compound. Then you can use a knowledge based scoring. In this case if you have the ligand and the protein, you can see the contacts and convert the contacts into potentials, right, and this potentials you can get the scoring function whether this can identify the actives, right, compared with the inactives. Then the consensus scoring, right, as we discussed in the secondary structure prediction, you check the different types of scoring functions and pick up from here and there, finally make the another scoring function, right, consensus from different functions. I will explain the details now how to do that. First one is the shape and chemical complementarity score. So, in this case either you can see the cavity of the protein site, the active site, and the ligand, you can see where this can fit, you can find the complementarity. So, in this case, so the protein surface, right, they are the active sites, you divide into various zones, and from this you can see how many hydrophobic interactions it could make, and whether the ligand side also the partner ligand we have that much atoms are available. Likewise hydrogen bond donor, how many donors in the protein side and how many acceptors in the ligand side, and so other way around, how many acceptors in the protein side and how many ligands donor acceptors in the ligand side. So, if we have the surface, right, we can divide the surface into several zones, and each zones because ligand is small one, right. So, whether this can fit with the binding site or not. So, you see the complementarity, what is how many hydrogen bond donors, if there is 10 hydrogen bond donors in the proteins and the ligand also there are 7 hydrogen bond donors, but no hydrogen bond acceptor, then can we fit? No, right, because if we have the hydrogen bond donor here, there we need the acceptor, so make the hydrogen bonds, right. So, they have different types of interactions, hydrophobic, hydrogen bond donors and hydrogen bond acceptors at the protein side, and get the same from the ligand side and see the complementarity whether these numbers will match or not. So, if they match, then you can say this ligand 1 is better than the ligand 2, right. So, likewise you can see the scoring based on the shape or the chemical complementarity scores, right, we can do that. And the second one is the empirical scoring, right. How the empirical scoring will work? Right, in this case if we get the binding affinity of known ligands, right. For example, if we have 100 ligands with this specific target, right. So, we know the binding affinity experimentally known, right. Then the right side we can calculate various parameters, for example, torsion, number of torsions, number of energy due to hydrogen bonds, energy due to the charges, energy due to the aromatic interactions and the other hydrophobic interactions. So, we can calculate various parameters, right, because we have the protein and we have the ligand, right, the affinity is known and the complex is available, right, then we can calculate all the parameters. So, when we get all the parameters, take the compound 1, right, you can see delta G1 this known, this equal to A into delta G torsion 1 plus B into delta G hydrogen bond 1 and so on, right. So, we take the compound 2, you can get the value delta G2, you can use this, right. So, now here if you see the right hand side, this delta G, all delta G's you can calculate from the complex, or if you take a free protein you can calculate, these all the values are known. So, what is the variable here, what is the constant here? The coefficients are common, they for example this A, B plus you can say constant, so that we do not know. So, here we know the delta G, this experimentally known, right. So, now fit these equations, for example, we have 100 compounds, you will get 100 equations, then how fit these equations? Using the principle of least squares you can do, right. So, if the principal least squares, you can fit these equations and you can calculate all these constants. Now, for any new compound, you can make the ligand and you can see the protein. When we make the complex, then we have all the constants, we calculate all the energy terms and fit, and then we will get the value of delta G. This will help you to score. If there are another 100 compounds or 200 compounds, we calculate all the energy terms and the constants we know. So, fit this equation, right, you can fit this equation to get the value for the delta G. Then we have 500 compounds, you get the 500 compounds, we get the delta G and you can rank. Oh, this is 1, this is 2 based on the scoring function, right. So, what are the advantages and disadvantages of having this empirical scoring? The advantage is very fast, because we need to calculate only this energy, energy terms, and you can also estimate directly, you can estimate the binding affinity, because we have the equation, we have this equation, get the values, substitute the values and you can get delta G, very simple, right. So, what are the disadvantages? Because when we, for developing this empirical scoring, we need the value for delta G, but if delta G is available for thousands of compounds, then you can get better results, but delta G is available only for few compounds, right. For example, if you take any specific target, you do not get many compounds, you get only few compounds. In this case, you can, the accuracy of getting this structures and binding, right, is difficult, because the fitting is not proper. Then you can see the discrepancy in binding affinities. Sometimes you have the binding affinity reported by different labs are different, in this case it is difficult to fit with this particular equation. Then also it depends on the placement of hydrogen atoms, because making the different interactions. So, it depends upon where the hydrogen atoms are placed. If it is wrongly placed, then we will have the issues with the hydrogen bond formation, right. Then also depending upon the transferability of the training set. Then the next one is there is no penalty time for the bad structures. If the structures are not good, even then if they interact, they make it interact, then in this case there will be a good score. So, these are the several disadvantages of having the empirical scoring function, but the good part is, if you get good number of affinity values and good number of structures are available, then you can easily get good score using this empirical function. So, this works fine for some cases. Then the next one is the force field scoring, right. What is the meaning of force field? Calculating different types of energies. So, here this is mainly this for the van der Waals energy, right, and this term this electrostatic. So, this is the major terms used in the Ampere force field, is widely used force field. So, this is well studied and also this is based on physical basis, because calculate the energy. Then you can see this energy and calculate the energy for any complex. You have the protein and you have the ligand, right. You can calculate the energy because we know the distance, right, and directly you can relate, okay, the energy is low, this is the probable compound, the energy is high, this is not a probable compound. So, for all the pairs you can calculate the energy, you can do it, it is easy. So, what are the disadvantages? Yeah, here it uses only some specific types of energies, for example, electrostatic and the van der Waals energies, right. In this case they may require the salvation as well as the other types of entropy terms, that is missing. So, we need to refine this force field, right, to get the better results. Sometimes the electrostatic is overestimated. So, in this case it will be difficult to rank the complexes. If the electrostatic estimated, then if there is the only positive negative charge atoms, then they will be ranked very high, like ignoring these hydrogen bonds and all. In this case we need to be cautious when you use this force field type of scoring. Second aspect is they also use various constants. The constants are also important because they derive from the known structures, right. This also plays a role when we get the non bonded interactions. So, we discussed about 3 different types. What are different types of scoring functions? Shape and chemical complementarity, empirical scoring function and the force field, right. Now, the next one is the knowledge based scoring function, right. The knowledge based scoring function, that depends upon how the atoms are in contact, right, in the ligand and the protein. So, what they do? So, they make a matrix, right, one side is for the protein, other side is for the ligand, and see the possibility of probability of contacts between two atoms. What are different atoms in protein?
CH.
N, C, O, N plus, O minus, S, right. Likewise, ligand also we can have the different types of atoms, and see how far the atoms are distributed. Take any space, say 3.5 angstrom or 4 angstrom, and within the distance they can see the pair preference, right, whether the ligands, right, there is a N and this N are in contact or not, N and O are in contact or not. So, they get this preference based on distance, right. Then you see the any specific distance, for example 3.5 angstrom, right. So, totally how many residue pairs, among the residue pairs how many residue pairs in different specific groups, right. We normalize the values and finally we can get this propensity, right. Take any distance, get the atom contacts, any pairs. So, based on the atom contacts we normalize the total number of contacts, this you can help to get this propensity. Then we convert the propensity, right, into the energy potential, right, using this equation minus RT logarithmic of this propensity. Because propensity is treated as a kind of partition coefficient, that means data from several sampling, right. Then this propensity is converted to the free energy using minus RT into logarithmic of propensity. So, now we can get these values, then we have the new protein ligand, we have the protein and the ligand, we can get the distance, right. For each pairs we have the delta G values and substitute the values and sum up the data and finally you get the score. So, now for any complex you can get this score and add up the scores. So, this is based on knowledge based scoring function. How this works? If any specific pairs of atoms they are preferred and that preferred atoms are also present in your new complex, then they will have high score. If it is not preferred, right, if there is highly preferred in this new complex, it will get low score depending upon the availability of these contacts in known protein ligand complexes, right, this will score the new complex. So, this is kind of empirical, but it is more general because it is more distance at the binding data, because we get more number of data, because binding data is not required, we need the complexes that data are available. So, we have plenty of complexes, we can do that. The disadvantages is, okay, they use typically a pairwise one, this is mean force, potential mean force, but the probability of the A and B to be in contact mainly depends upon the surrounding atoms, right. In this case difficult to map, right, all the contacts in the case of the protein ligand complex. So, second one is the hypothesis originates from the statistics of the uniform liquid one, but here this is totally non uniform medium, right, in the case of the protein ligand complexes, right, in the environment. Then the next one is the consensus. So, now we have a different scoring function, some scoring function performs good in some aspects. So, they try to integrate multiple scoring functions to get a consensus score, either they use all the scoring functions or they use the data from different scoring functions with different weightage, right. This will see there can be any better than any single scoring function for predicting this binding affinity. So, there are various scoring functions we discussed. What are the various scoring functions?
Shape
complementarity and chemical complementarity, empirical based scoring function, force field based scoring function, a knowledge based scoring function, and the consensus. How the consensus works? Why the consensus is important?
It sums all this.
Yeah, because the rationalists they think if you take any scoring function, they give you some data, but if you compare, that could be better than you are using any of the single ones. So, in this case they try to use experimental data and see the different scoring functions and they merge the scoring functions, either they use all the scoring functions or take some information from different scoring functions and finally they try to get a consensus one, right. In this case you can get the better scoring function, right, so that it can account the binding affinity of various protein ligand complexes, right. Now, when using this such sampling space and the scoring functions, right, we can use to screen the compounds, that is called virtual screening, we will discuss in this class, right. So, get the compounds. So, there are various scoring functions and the software available in the literature, right. For example, force field based software, the gold score, the dock and autodock, right. Autodock is the widely used public domain program that is available for free in the academic usage. Then there is some empirical based force field like the chem score or the PLP or the glide extra precision, right, or standard precision, and knowledge based potential like the PMF or drug score or ASP and so on. So, several software available in the literature, right, and glide is the commercial one and the autodock is for free, right. You can use any of these software, right, to get this docking with the protein with any ligand. Then once we have the docking, then if we have a set of compounds, right, for example, the NMN there are 2 million compounds and the zinc 35 million compounds, you can use this information to pick up the probable ligands, right, which can be a potential lead compound for in the case of drug discovery. So, in summarizing, what did we discuss in this class?
Computer aided.
Right. So, what are the various aspects of this computer aided drug design, right. So, if we have the what are the information required for this docking?
Binding site.
We need the protein site as well as the ligand, right. So, and then if you have this one, right. So, we have two types of docking we discussed, one is the rigid docking and the flexible docking. What difference between rigid docking and flexible docking?
Both are.
Yeah, rigid docking protein site is rigid, ligand also we can make conformation and then try to dock, right. In the flexible docking it is proteins have different conformations, right. In this case you can generate various conformation, right, which will take time.
Flexible.
Flexible docking, right, because we have searched through various conformations, right. So, what are the difference two different major aspects in docking?
Search algorithm.
Search algorithm
Scoring.
and the scoring function, right. So, what are the various search algorithms we can use? Systematic search and the stochastic search. What is the difference between systematic and stochastic search? Yeah, it takes all systematic we have to do, but it is time consuming, right. Stochastic it can randomly sample, right, and you can get the probable scoring, right, the pose, right. Then the energy functions, what are different types of energy functions?
Scoring.
So, scoring functions, right, different types of scoring functions based on the complementarity or the empirical, or you can see the energy or the using the propensity values and the consensus ones, right. So, we have the sampling and then we have the scoring function, then you can dock the compounds, and if you have a set of compounds in library, you can do the virtual screening, right, to identify the probable compounds as the potential lead compounds, right. In the next class I will discuss about the virtual screening of compounds, right. If we have a set of compounds, how to identify the best ones, right, and to get the probable compounds which are for example lead compounds, right. And then we will see the how the quasar models are also used, right, to identify the novel potential inhibitors in the next few classes. Thanks for your kind attention.